So 78, a patient has disturbed absorption of fats hydrolysates. It might have been caused by deficit in the small intestine cavity. Let me define these fat hydrolysates. It's these hydrolysates actually help in the breakdown of fats to become fatty acids. Then for bile, we have cholesterol catabolism. By lipid absorption. And then for bile pigments, these are pigments, of course, these are like substances that are being released after hemoglobin degradation or heme degradation. So substances formed after heme degradation. And a typical example is I think they call it bilirubin, something like this. I'm not sure of the spelling.
So, what did you write here? Substances formed, then what?
Sorry,
What's the word after formed?
after.
Oh, okay.
After I think bilirubin, just bilirubin. And then what again here? The polytic enzymes are well, let me skip that for now. Liposoluble vitamins. These are just vitamins that can dissolve in fats. In fats, lipolytic enzymes. Well, of course, enzymes that participate in the breakdown. In hydrolysis of fats. In this case, sodium ions. I don't know. Are they well? Okay. At least concerning this question, these definitions go. So a patient has disturbed absorption of fats hydrolytes. It might have been caused by a deficit in small intestine cavity. Um, this is a clear picture of absorption of fats or fats breakdown. So if this is the intestine, sorry, let me just say this is fats. Well, fats. The liver itself secretes bile. There's the gallbladder and also the bile, just here in green. So it is what is being secreted as bile acids. I'm drawing this just so that you remember the anatomy and physiology of this. So this is what goes down to break or cause fat emulsification. Breakdown. Okay, so this is a clearer picture than this pathway that you have here. So um, this is the fats, the same thing I drew here. And then you have emulsification to form all these other components, which is the triglyceride and then further broken down stuff like that. So this bile salts you are seeing here is also termed the bile acids. So I'm going back to our question now. We look at the closely related. Patient has disturbed absorption of fat hydrolytes. The absorption is here already. It might have been caused by deficits in small intestine cavity. So the bile acids, which can also be um, what can they use here? Bile salts or bile acids, yeah. Or bile salt. In case you see bile salt, it also means the same. It refers to the same thing concerning this action, concerning the function rather.
Yeah, but how did we conclude from the question that it is bile acid? Is it just because of the fat we concluded?
Lipid absorption in that whole process. Where is it? Here. If you're talking about bile pigment, this is just a substance that is being formed after hemoglobin is broken. I'm talking about option two now. So that is not supposed to be the answer. Do you understand that?
Yeah.
Then for lipolytic enzymes, these are just enzymes that take part in the hydrolysis, but they are not really concerned with the absorption of the lipid. Hydrolysis is also breakdown. So in addition to the action of absorption, or in addition to the breakdown, there's absorption, which is being done by the bile acids or bile salts. That's what I'm trying to differentiate here. And then this last option are just vitamins that can also be dissolved in fats.